Hello everyone. Today we are going to discuss about the glyoxysomes. Glyoxysomes are the peroxisomes of plants, or we can call them as microbodies as well. They occur in the cells of yeast. Neurospora, and their number is high in many higher plants which are having oil-rich seeds. These glyoxysomes resemble with peroxisomes in their morphological character, except that their colloidal core consists of dense rods of 6.0 micrometer diameter. They are having enzymes for fatty acid metabolism and glyconeogenesis. These enzymes are responsible for or conversion of stored lipid molecules which are present in the spherosomes of germinating seeds into the molecule of carbohydrates. So if we will talk about the functions of the glyoxysomes, these glyoxysomes are involved in basic two main functions: fatty acid metabolism, beta oxidation of fatty acids, and the another is the glyoxide cycle. So first we will discuss about the fatty acid metabolism. During germination of these oily seeds, the stored lipid molecule which is present in the spherosomes are hydrolyzed by enzyme called lipase, and there is also the presence of phospholipid, and this phospholipid hydrolysis is carried out by the phospholipase enzyme. The long chain fatty acids are released by these hydrolysis, and by the beta oxidation there is the successive removal of 2 carbon fragments in each process of beta oxidation. So if I will talk about the beta oxidation process, like you can see here, when we are having the fatty acids, first the fatty acid is, we have to activate this fatty acid by the addition of CoA. So fatty acid, in presence of fatty acid thiokinase, is converted to fatty acetyl CoA. Fatty acid is converted to fatty acetyl CoA. Then fatty acetyl CoA undergoes oxidation process in presence of electron donor as a FAD, then converts it into the trans 2 enoyl CoA. So fatty acid is converted to fatty acetyl CoA by the activation reaction. Then fatty acetyl CoA, in presence of enzyme fatty acetyl CoA dehydrogenase, is converted to trans 2 enoyl CoA. Then this trans 2 enoyl CoA goes hydration reaction to form the L3 hydroxyl CoA in presence of enzyme enoyl hydratases. So trans 2 enoyl CoA is converted to L3 hydroxyl CoA by the enzyme enoyl hydratases. Then this L3 hydroxyl CoA is converted to 3-ketoacetyl CoA by the oxidation process. This oxidation process is carried with the help of NAD by the enzyme L3 hydroxyl CoA dehydrogenase. L3 hydroxyl CoA dehydrogenase. Then this 3-ketoacetyl CoA is converted to acetyl CoA, and the remaining chain of the fatty acid. This is the thyrolytic cleavage, which is carried out by the beta ketothiolase enzyme. So, in plants, beta oxidation is carried in the glyoxysomes as well as in mitochondria. But in plant seeds, beta oxidation occurs only in the glyoxysomes. In plant cells, the acetyl CoA, the product of beta oxidation chain, is not oxidized by the Krebs cycle as we have seen in the mitochondria, because it remains partially separated from the enzymes of Krebs cycle. Instead of it, acetyl CoA undergoes the oxalate cycle to convert into the succinate. Now, the another we have to discuss about the glyoxylate cycle. So, it involves some of the reactions of Krebs cycle. Like the first reaction is the acetyl CoA interacts with the oxaloacetate in presence of citrate as an enzyme to form the citrate. Then this citrate is converted to isocitrate by aconitase. Then isocitrate is converted to glyoxate and succinate by the isocitrate enzyme. These are the similar reactions which are carried in the Krebs cycle. Then this isocitrate is converted into the glyoxylate and succinate. So, the glyoxylate, the another molecule of acetyl CoA, forms a molecule of malate by malate synthetase. Acetyl CoA plus glyoxylate gives the malate with the help of the malate synthetase. This malate is converted to oxaloacetate by the malate dehydrogenase for the cycle to be completed. Thus, we can say the overall reaction of the glyoxylate pathway involves two molecules of acetyl CoA plus NADH gives the succinate plus NADH and plus H positive ion. Succinate is the end product of glyoxymal metabolism of fatty acid and is not further metabolized by this process. It is involved in the glyconeogenesis. This succinate, which is there in the cell or in the glyoxysomes, is used for glyconeogenesis process. So, the succinate is converted to oxaloacetate in the glyconeogenesis process. Glyoxysomes do not contain the enzymes fumarase and succinic dehydrogenase. The two molecules of oxaloacetate are formed from 4 molecules of acetyl CoA without any carbon loss. This oxaloacetate is converted to phosphoenol pyruvate by the enzyme phosphoenol pyruvate carboxylase and the loss of 2 carbon dioxide molecules. So, the phosphoenol pyruvate is converted either to the monosaccharide, example glucose or fructose, or this phosphoenol pyruvate is converted to disaccharide, that is sucrose, or we can say polysaccharide, these are the starch. So, phosphoenol is first converted to triose phosphate, then triose phosphate is converted to fructose 1,6 diphosphate. Then fructose 6 phosphate is directly converted to fructose, or this fructose 6 phosphate is converted to glucose 6 phosphate. This glucose 6 phosphate can relieve glucose, or they can undergo another chemical reaction which form glucose 1 phosphate. Then this glucose 1 phosphate can form ADP glucose, then finally leads to the formation of the starch. Or glucose 1 phosphate is converted to UDP glucose. Then with this UDP glucose then interacts with the fructose 6 phosphate, which forms the sucrose molecule, This is the overall involvement of glyoxylate in the plants. How the glyoxylate enzyme are responsible for the beta oxidation process and the gluconeogenesis process.